Today we will be talking about epistasis. Epistasis is when two or more genes contribute to the same phenotype. There are 6 modes of epistasis, but we will be focusing on only one mode today. To start off, we will complete a dihybrid cross between two heterozygous individuals, both sharing the genotypes big L, little l, big D, little d. A dihybrid cross is a cross between two individuals and two different traits. This cross gives us a 9:3 to 3:1 ratio, with 9 individuals having a dominant allele of both genes, 3 individuals being dominant for L and recessive for D, 3 individuals being recessive for L and dominant for D, and 1 individual being homozygous recessive for both traits. However, this is not always the case, and things change in nature. The variation is a result of epistasis, and the mode of epistasis we will be focusing on today is single dominant epistasis. Single dominant epistasis is when the dominant allele at one loci masks the expression of an allele at a different locus. To explain, we will be going into the M&M world and we will be observing their population. As we can see, the M&Ms of this region are all warm colors, but the majority of them are orange with a few reds sprinkled in and only a couple of yellows. Let's take a look at the epistatic pathway to understand why the population looks the way it does. The precursor color for the M&Ms is orange, which makes sense since majority of these M&Ms are orange. Enzyme L for lightness and enzyme D for darkness play a role in the colors of these M&Ms. Let's take an M&M that is ready to be colored and find the possible colors it could be. Following the pathway, being dominant L inhibits the M&M in being any color other than orange. The blue M&Ms represent the security guards at enzyme L. Being dominant does not give you a pass to the yellow, therefore the security guards will stop the M&M at orange. However, if the M&M has 2 little L's, therefore being recessive, it can be either yellow or red. Being homozygous recessive L is your pass to the security guards into yellow. Enzyme D then plays a role in determining if the M&M will be either red or yellow. Enzyme D is covered by green security, and unlike blue security, the key to pass through is a dominant allele. If the M&M has a dominant D allele, then the M&M can pass through security one more time and it will become dark and red. If the M&M is homozygous recessive, little d, then it cannot pass through the green guards at enzyme D to become dark and will have to be yellow. This brown M&M has 3 different colors that it could be. If the genotype for the M&M is big L, big D, little d, at the end, it will become orange. There is a higher chance that it will turn out to be orange since blue security is super strong, which is seen in the overall population of the M&Ms. Using the ratio, approximately 75% of all M&Ms will be orange, 18.75% of all M&Ms will be red, and 6.25% will be yellow. We can recreate these probabilities through a cross. The dihybrid cross to the left shows the ratio when 2 heterozygous M&Ms are crossed, the ratio being 12:3:1. At the bottom left, a cross between big L, big D, and little l, big D, little d is shown. The epistasis is not shown here since there's only 2 options that the cross would allow. The homozygous dominant L alleles would make the M&Ms orange. The homozygous recessive L alleles would make the M&Ms red since there can't be homozygous recessive D alleles for the M&Ms to be yellow. I hope this helps you better understand single dominant epistasis.